Okay, I hope that everyone can hear me now and that we are online. So very good evening and a warm welcome to each of you joining our tonight's Ringvorlesung. For those of you who don't know me, my name is Roland Weierstein-Pust. I'm professor for clinical psychology and psychotherapy at the medical school in Hamburg. And I'm also director for research and development at the Oberberg Group. So if you're joining our Ringvorlesung for the first time today, just let me say a few words about this format so that you get an idea what's happening today and why are we offering this Ringvorlesung. So the Ringvorlesung is a lecture series that has jointly been developed or organized by the medical school in Berlin, the medical school in Hamburg, and the Health and Medical School University at Potsdam. And so what is our mission? So every term we try to bring together researchers from different health sciences presenting the latest state of the art in their respective fields. And all this is done under the umbrella of health and social participation from an interdisciplinary perspective. Okay, so this is very important for us, and I also hope that tonight not only medical doctors and psychologists are attending this lecture, but that we also manage to inspire researchers, students, and participants from other disciplines to also be part of this lecture tonight. This winter term, this was our idea to come up with an overarching theme that is called the epigenetics of stress and trauma from research to practice. And you see, for me, it's very important that we come to the point where we can bridge the gap that still exists between practice and research. And I know that there are some of you are more interested in the practical field and asking the question of how the fundamental research that will also be presented in this talk and also the next presentation, how this then transfers to the application in clinical practice. But I think that the presentations with the two with which we will start will probably let you get prepared and develop a better understanding of the two presentations that will be follow in January and February next year, where we will get insights into the epigenetic mechanisms in trauma survivors in the context of Rwanda and in South Africa. But I think today, also with the presentation of Professor Mansuy, we will get in the fundamental basis that we need in understanding how epigenetic works, what epigenetic research brings us, and how epigenetic mechanisms also will help us to understand how trauma and stress manifest in the individual, and also what we will learn from today's talk, how this will also be transferred from one generation to the other, maybe through the germline. And I hope that this in the end will give you a more holistic perspective on this topic and hopefully inspire both researchers and practitioners to come together, to elaborate on this field and sit together and see how we can then also strive towards our maybe ultimate goal of improving health and social participation. For those of you who are also interested in the CME credits, you have maybe also seen it already in the chat that if you need to get the CME credits, you have to send an email to Winke Ladentin, because for the credits you will also have to leave your address, and then this will be sorted by her. As you might have also recognized this term, and this is new in this term, that this lecture all the lectures will be in English, and but I hope this won't prevent you from engaging in discussions. So if you have any questions, if you want to learn more about certain aspects, also from the results that Professor Mansuy is presenting, just feel free to use the Q&A section. So I think it's my intention is that today we have a great time, we have time to exchange, we get inspired, and no one is here to judge you. So even if you're coming from the clinical practice and you're not fluent in English, you don't know how to choose the right scientific expressions, don't worry about it. We won't judge you. So just type in your questions and we will translate it for you. You can also try it in English, feel free. Well, but now let me come to introduce our tonight's speakers, Professor Isabelle Mansuy. For me personally, I feel very much delighted, Isabelle, that you accepted my invitation to give a presentation on your work. And I just when I was thinking back at the days I was working in Constance, it's more than 10 years ago, I think, or when Professor Mansuy and I met for the very first time, and I have to say that I've been very inspired by your work, because I think it provides new insights that also have had a significant impact on my understanding of how I think about stress and trauma and transmission of trauma from one generation to the other. And I think one of these compelling advantages your research has is that the mouse has a different life cycle than a human, and you can study things or understand mechanisms that you cannot derive from work in humans. And for those of you who do not know Professor Mansuy so far, she's professor in neuroepigenetics at the medical faculty of the University of Zurich and the Department of Health Science and Technology of the ETH in Zurich. She's trained as a neurobiologist, and she has conducted research on molecular mechanisms of memory for 20 years. And the research why I've also started a request and asked her to give a presentation for us today is that she has also switched to the novel discipline of epigenetic inheritance and is one of the pioneers in this field. And as I have also tried to mention, her findings contribute to the paradigm shift in biology and an integrated understanding of epigenetics into the notion of heredity. And I think by this talk today, we will get really fascinating insights. Of course, I have to say that Professor Mansuy has authored more than 150 research articles, reviews, and book chapters in neuroscience and neuroepigenetics, and her publications involve all of the most famous research media that you can imagine. There's also a book that she has published for the lay public that we can recommend to you. So if you also started to think, what can I buy for Christmas as a present for my family, or what is that I want to wish to have as a Christmas present? It's called Wir können unsere Gene steuern. So it's a very interesting book, and I can highly recommend it to you. And Professor Mansuy, she's an active member in multiple scientific and research funding boards, for example, the Swiss Academy of Medical Sciences, the European Academy of Sciences, and the European Molecular Biology Organization. And what I think is also quite fascinating about her, that she's a knight in the National Order of Merit and holds the Legion of Honor in France. And to give you an idea of how famous not only her work is, but also Professor Mansuy as a researcher, that she's also nominated for the Nobel Prize in Physiology or Medicine. So Isabelle, it's a very, very big pleasure for me to welcome you in the name of my colleagues, all our guests today, and the audience. And I'm very sure that it will be a great time listening to you. So
thanks very much, Roland, for this very nice introduction and for the invitation, the offer to talk to this audience tonight and present the concept of epigenetic inheritance and our work in the field. I'm sorry that I cannot be there in person, and it's a little awkward because I just have my screen with my talk. I don't see anyone, so I'll try to look as if I was in front of you. So what I'd like to do tonight is first start with a definition about the concept of epigenetic inheritance. What is it? It's a kind of a new way to think about heredity. But it's very justified, and it's something that is involves us and our life quite prominently, but which mechanistically speaking is still not well understood. And then I will show you how we have approached the question of the mechanism of this form of heredity using a mouse model with a little bit of analysis in humans. And I will show some of the data we collected, which suggest or which identify some epigenetic factors in sperm cells, which are likely responsible for the transmission of exposure to traumatic experiences across generations. So there are many factors which influence our body and our brain, I mean, throughout life, from the very beginning, even before birth, at birth, during childhood, adolescence, adulthood, until late old age. Whether food or nutrition, whether good or bad, life experiences, traumatic experiences, stress in special circumstances like during war, but also in our own daily life, violence or abuse in adulthood or in childhood. Drugs and chemicals, cigarette, alcohol, medicaments, but also environmental pollutants, toxins, endocrine disruptors, for instance, which are everywhere in our environment, in the plastic, in the pesticides used in agriculture, in many of the things which surround us and are in our house. But not only negative factors, but also positive factors can influence our body. And what's important to realize here is that every single cell in our body, whether brain cell, heart cell, or germ cell, can be modified, can be changed, not in the genome, not in the DNA sequence, but in the epigenome, not in the DNA sequence, because the DNA sequence, I mean, it can be perturbed, it can be broken, but by UVs, carcinogen, X-rays, I mean, this kind of very violent things for the cells, for the DNA, but poor diet or traumatic stress is not going to break or to induce any genetic mutations, any mutation or changes in the genome itself in principle, but only in the epigenome. So the question is this, the concept is this, that there are all these environmental factors or life experiences, whether in early life or adult life, can change the epigenome in every cell in our body. And indeed, now if we talk about quite severe exposure, now I'm not. It doesn't mean a light exposure. For instance, if you eat McDonald's at some point, it's not going to affect your body for the rest of your life. Here, exposure is chronic exposure, something very severe. If we are talking about stress, that would be stress in childhood, for instance, which undergoes for weeks or months or even years. And high-fat diet would be a lifestyle. People who eat only or this type of diet. So now the notion of transmission. So the idea is that being exposed to any one of these factors, sometimes they are even combined, can affect the epigenome in our cells. And if the epigenome of our germ cells, in this case sperm cells, are affected, there is a certain likelihood that symptoms induced by this exposure, for instance, metabolic disease for high-fat diet or psychiatric diseases for traumatic stress, that these symptoms may be manifested by the progeny, by the children. Because if there are changes in the epigenome in any of these cells, including in sperm cells, epigenetic changes, and these epigenetic changes persist until conception, then they may carry a memory of exposure, a molecular memory of exposure, which may recapitulate symptoms, some symptoms, not necessarily all symptoms, but some symptoms in the progeny. Now, if it's a pregnant mother, pregnant woman who is exposed, not just her baby, but also the future gametes of her baby. So it's not only one generation, the two generation, but also third generation. And it's it's easy to understand that if these gametes here, which may be only progenitor germ cells, but if their epigenome is very severely perturbed and these perturbations are not corrected during development, they could still keep a trace of exposure. Now, what about following generations? Is it possible that the germline, the epigenome in germ cells, whether male germ cells or female germ cells, can they be altered persistently in a way to explain the transmission of some symptoms across multiple generations? So it's a concept which requires to completely change the way of thinking about heredity. Classically, heredity is in the gene. you receive genes from your mom, from your dad. They are mixed, and that's your that's your genome. What makes your identity and your personality. But now the idea of epigenetic inheritance is that it's not just the sequence of the DNA, but this is also the epigenetic factors which are on the DNA, like DNA methylation, or around the DNA, like non-coding RNA. And these factors may also carry, they do indeed carry memory or signals or changes which are induced by exposure and which are can be transmitted across generations. So this notion of epigenetic inheritance has been observed across multiple species, actually, quite broadly. In C. elegans, some things like exposure to temperature can induce phenotypes up to the 14th generation. In rodents, exposure to stress, to paternal obesity, to some epigenetic factors or endocrine disruptors, they have also been shown to have to induce some symptoms, whether metabolic symptoms, behavioral symptoms, or different types of clearly identifiable manifestations that can be transmitted across multiple generations. In humans as well, although the epigenetic aspect of humans is still not proven yet. But when we know from historical cohorts, like the people who were exposed to famine in West Holland at the end of the Second World War, that the descendants across at least 3 generations of women who were exposed to this famine, they have increased cardiovascular diseases, reduced longevity, and they have a number of signs of exposure. So there are a few cohorts like this. No, I don't have more detail about that, but in terms of stress, there is a study. Well, there is the work of Rachel Yehuda, who is in the US, but she's a descendant of Jews who flew to the US during the Second World War, and she has spent all her career, now almost 30 years, if not more, on looking at the consequences of Holocaust exposure in people directly, the victims and the children. And she could really show that in terms of psychiatric disorders, the incidence of psychiatric disorders, but also other disorders, is increased in the children of the people who have been exposed and who survived. Rwanda genocide has also been suggested or implied to induce symptoms, psychiatric symptoms, which are transmitted from the exposed mother to the children. Now, in psychiatry, it's very important because it's extremely relevant. It's known that traumatic events like physical, sexual violence, abuse, abandon, or neglect experienced in childhood or adolescence can induce or cause or be responsible for a number of psychiatric disorders early in life or later in adulthood, including bipolar depression, antisocial behaviors like borderline personality disorder, cognitive and personality disorders, schizophrenia, and in some cases can even be responsible for suicide. And that's many of these diseases, they run in families. But despite a large number of genome-wide association studies, I mean, there is not a single gene which is responsible for these complex diseases, but there is a genetic susceptibility which, in combination to exposure to traumatic experiences in early life in particular, it could be sometimes in late life, in adulthood also, but trauma, physical and very often sexual abuse in childhood are very often associated with severe, psychiatric pathologies, which affect people across their life and across families. Now, how to look at the underlying mechanism. So to do this, we developed a mouse model of early trauma many years ago, really based on the human literature. in particular borderline personality disorder literature, because it's a disease which is quite complex. It's a combination of depression with antisocial behaviors, problem with memory, risk-taking, and very often, most of the patients have been exposed to traumatic stress in their childhood. So using this literature, we developed a paradigm which is a combination of maternal separation in mice. So here you see a mother with her newborns. Well, they are no longer newborns because newborns are pink. They are a couple of days old. But what we do is that one day after birth, we separate the pups from their mother for 3 hours a day, anytime during the day, every day, for 2 weeks. So from P1, from one day after birth till the 14th day of life. And during these 3 hours of unpredictable maternal separation, we also stress the mother unpredictably, anytime during the 3 hours, either on a tube like you see here, or in cold water, forced swim. So the combination of the maternal separation, unpredictable maternal separation with unpredictable maternal stress is an extremely severe stress. It's abbreviated MSUS. So we use this paradigm. So again, every day for 3 hours from postnatal day 1 to 14. And then the last week before weaning, we wean the animals at 21 days of age. The animals they grow normally with their mother, so it's only 2/3 of the pre-weaning period of exposure to this stress. Then we wait, after weaning, we wait until they are adults, and we do lots of testing on them, but we also breed them. So we derive patriline, as you see here. They are the traumatized males. That, I'm sorry, I'm going to use the term traumatized males is just to make it easier. It may, for those of you who work with human, it may sound a little too anthropomorphic, but I'll allow myself to use this term because it's just easy to understand. So these males, we breed them with controlled wild type naive females to generate a second generation, and then we repeat this breeding with each generation fresh females, naive and unstressed females, and we have now generated a cohort until the 6th generation. So these manipulation here plus the breeding to at least F2, we did 34 times. So in the past 20 years, we generated 34 cohorts, and we are going to start the 35th cohort very soon. So our data, we have confirmed that this paradigm is really consistently inducing various behavioral alterations and physiological metabolic alterations. To come back to the notion of direct exposure, you realize that here these pups are directly exposed and the germ cell as well. So this generation here comes from a germ cell, let's say a sperm cell in this case, which has been directly exposed. So if those animals have symptoms, you can somehow relate it to the fact that the sperm cell they come from has been exposed. Now it's no longer the case for the further generation, F3, F4, F5. Not a single cell, not even the sperm cell they come from, has been exposed directly to the trauma. So if they show symptoms, it means that there have been molecular mechanisms or molecular factors which were maintained in sperm cells or reinstalled at each generation, which still carry a memory of exposure and are responsible for the symptoms. So what are the symptoms? I will quickly show you a few of them. So the animals have depressive-like behaviors. So to test depression or depressive-like behaviors in mice or rodents or rats, there are different tasks. This one is it's the forced swim test. You place the mouse in this container of cold water, and a mouse will swim and try to escape. A depressed mouse will float. If you inject this mouse with an antidepressant, it will continue to swim like this one and try to escape. Some people argue that, well, maybe they are smarter and they start floating because they understood that there is no escapable way. Perhaps, but the fact that it's reversed by antidepressant suggests that it's really more depressive-like behaviors. Learned helplessness. So we tested the animals on this task, but we used other tasks which also assess this type of behavioral despair. And what we observed was that if you look at the time spent floating, you see that it's increased in the MSUS mice. So those directly exposed is one, their progeny and the progeny of the progeny. But afterwards, F4, F5, there is no longer any significant depressive-like phenotype. So depression or depressive-like phenotypes are induced in this animal at least until the 3rd generation. And indeed, here again, this generation has never been exposed to any stress. This one has sperm, but not this one. So it suggests that, I mean, there are probably epigenetic factors going on here. Another task is the elevated plasmase. So it's an elevated plasmase, as you see, it's elevated 80 centimeters from the ground. And you see this mouse is placed initially in the center of the plasmase, and it will be allowed to explore, and a mouse will normally explore these open arms only after checking the danger, doing a lot of risk assessment. So it's normally used to test anxiety, but you can also use it to assess risk-taking. And what we have observed in these mice, if we look at the time spent in the open arms, the arms are about 80 centimeter, 60 centimeter long. So it's quite dangerous because it's high on the floor and the animal are not protected. But what we observed is that the mice which are exposed directly to the stroma and the progeny down to the 6th generation, they show increased risk-taking. So they spend much less time assessing the danger, and they spend more time on the open arms, and they step in the open arms faster than the other one. So risk-taking behaviors, which is very consistently transferred across generations through the patriline. We don't know about the matriline till F2, but we have not tested the matriline further down. All the data I'm going to show you today are from the patriline, so the males which are bred. Social interactions are also altered. As you see here, it's a discrimination ratio between novelty and remembering the peer, and social interaction is altered till the F3. We have not tested F4 actually. And this is indicative of the animals not being able to read social cues. They have proper olfactory function, so it's not a problem of not identifying the other animal, but it's just recognizing and showing recognition, because if we place them repeatedly together, the amount of time spending investigating or looking at the other decreases because they know each other. So our mice, they don't do this. Each time they see the same mouse, they need to explore the mouse because as if they have never seen it before. So it's a problem of reading social cues and using social cues for social recognition. They have impaired memory, actually. So in addition, there are many behaviors which are altered. I won't show you everything, but memory is, as I said, quite severely impaired, object memory and field memory. But in addition to behavior, metabolism is also quite profoundly impaired. You see here, glucose is decreased. So this is in controls and in MSUS mice. This is in the 2nd generation. And if we stress the animal, you see that the response to glucose increases and then comes back to baseline. So the mounting and recuperation of the response is normal, but the level of glucose is always lower in these animals, in F1 and in F2, and down to F4. It's a little less pronounced, but there is a decrease in the amount of glucose and also in the amount of insulin, actually, in blood across generations in these mice. And metabolism is not only altered in blood, but it's also altered in brain. So this is here the result of a mass spectroscopy. So it's done in vivo here in the 2nd generation. And you see that there are multiple metabolites, including choline, I think, and here glutamine, which are significantly altered. And aspartate as well is significantly altered. So in the brain of the offspring of males which have been exposed to the trauma. So metabolism in blood, in brain is quite severely impaired. So now you understand that this trauma exposure affects a lot of things in the body. indeed, we looked at cardiac functions, and we found that the heart is hypertrophic. We looked at the third generation across age, and there are dysfunctions in systolic and diastolic dysfunction, indicative of susceptibility to do for heart failure in these animals. So the heart is affected, the blood is affected, bones are also affected. So wherever we look, basically, we see differences in these or alterations in these animals, some of which persist across generations. So what could be the mechanism in germ cells, in sperm cells, that could explain these symptoms? We know that the transmission goes through sperm cells because we used different strategies: in vitro fertilization, artificial insemination, and the females that we use are naive, they are good mothers. So we use all these strategies to exclude all of the factors: maternal care, behavior of the father. The father never sees the pups. There is breeding for a couple of days, and then the parents are separated, and the mother delivers 3 weeks later. So the father cannot communicate to his offspring because he never sees them. So we know that there are mechanisms or factors in germ cells, in sperm cells, which are responsible for the transmission. So what could they be? So on this slide, indeed, you see it's a very simple scheme of what germ cells, sperm cells contain. Of course, they contain the DNA, which is packed and protected. The chromatin looks a bit different than in somatic cells. You see most protein histones are replaced by protamines to be able to compact the chromatin more than in somatic cells. But DNA, despite having another type of configuration, can be methylated. So there is lots of DNA methylation in sperm cells. But there are also modifications on the protamines and modifications on the remaining histones in histone proteins. In human, there is about 15% of histone proteins which remain. But in addition to these classical epigenetic factors, so DNA methylation and modifications of protein histones, there are many non-coding RNA in sperm cells. Small, long, medium size, so there is a whole population of non-coding RNA and also messenger RNA in these cells. Messenger RNAs which come from previous stages because of development, because sperm cells themselves, they are transcriptionally inactive. Their job is just to transfer the genome and the epigenome from the sperm to the oocyte during fertilization. But there are still messenger RNA in them because the prior stages like spermatocyte or spermatogonia, spermatocyte, spermatid, there is lots of transcription activity, and some of these mRNA are degraded, but some are maintained. So the RNA population in sperm cells is huge. So it's something that we have looked at. So we did a little bit of work on DNA methylation, on histone modifications, yes, but it's not easy to identify and to quantify these modifications in sperm cells. So we focused our work in the past years on RNA. So the idea is that the RNA load of sperm cells in males which have been exposed to trauma is modified, is altered. So is it the case? Maybe I just summarize the concept again. So the concept is after exposure, so any cell in the body can be altered in the epigenome, so DNA methylation, histone modification, so non-coding RNAs, or other factors that we have not looked at or thought about looking, so in neurons, and the same changes or different changes could also occur in sperm cells or in oocytes if it's a female. And that if these modifications, altered RNA populations, altered DNA methylation profile, or altered histone modifications, they are maintained, they could eventually explain the transfer across generations. So now, to come back to RNA, we looked at whether RNA, small RNA or long RNA, is altered in the sperm of males which had been traumatized. So the males are exposed to trauma when they are 1 day old, from 1 day to 15 days old. This is the window of exposure, 2 weeks. And then nothing happens to them, and we check we collect the sperm when they are adults, so many months afterwards. They are maybe 5 months or 6 months of age, and we did RNA sequencing. And you see that this is the controls and MSUS. So each lane here is the pool of 5 sperm, the sperm of 5 males. So you immediately see that there is a difference. There is a lot more yellow. Yellow is more for overexpressed. So there are many microRNAs which are in excess in the sperm of MSUS, but not just small RNAs, but also long RNAs. Some are overexpressed and some are downregulated. So we have a whole list of these RNA, and they include messenger RNA, but also non-coding RNA, including transposon RNA. So clear, the RNA population in the sperm of these males are severely altered. Now, is it the case in human? So we had access to sperm from men that are in Pakistan. That's it's the work of a former PhD student in my lab, Ali Javaid, who is Pakistani himself, and who proposed to collect samples from children. I'll show some of this later to you, and from men in Lahore, Pakistan. So these men, they are going to a diagnostic lab, which is called the Chuktais lab, for giving this semen for virus analysis. And what Ali managed to do is to establish a collaboration with these labs, diagnostic labs, and have the possibility to talk to the men who were coming to give their semen, ask them if they are interested to participate into a study with us, and ask them to fill out a questionnaire, a few questions. I think there were 5 questions about their trauma in childhood. So whether they were exposed to trauma before the age of 17 and how many types of trauma. And then upon their agreement, we obtained an aliquot of the semen that Ali sent back to us here in Zurich, and that we analyzed using exactly the same protocol as in mice. And you see that we focused on a few microRNAs that we knew to be altered in sperm, in blood, in brain of males exposed, of males exposed to trauma when they were pups. And we could indeed see that the same microRNAs appeared to be altered, like 16 microRNA 34C, 375, 449. There were, these two actually have been shown by another group, by the group of Larry Feig, in another completely independent cohort of stressed males, to be altered in sperm. So you see that there also seems to be a downregulation, which is proportionate to the number of trauma, with 2 trauma before the age of 17 being more severely decrease. Now, this is correlative, right? We have these mice exposed to trauma, which have an altered RNA population in sperm. We have men exposed to trauma when they were children, who also have alteration in microRNAs. So, but what is the evidence that it's the mRNA or the RNA in sperm which is responsible for the transmission? So to do this, you need to mimic these changes. Experimentally, and see whether reproducing the alteration in RNA in sperm and then obtaining an offspring would result in symptoms in the offspring. That's what we did using a micro-injection approach. So we have here an oocyte. So it's an egg which has just been fertilized. It's control egg. And we injected a control egg, which is going to give rise to an embryo, a mouse embryo. And we injected these eggs with RNA, total RNA, extracted from the sperm of males which had been traumatized. And we have always controls where we take eggs like this also, but we inject them with control, with the sperm of control males which had not been stressed. And then after they are injected, the eggs are transferred by surgery into mothers, which are prepared hormonally. And then we wait until they give birth 3 weeks later. And these animals, we wait until they're adult. And then we tested them. We tested their behavior. And we also bred them to obtain an offspring. So these animals come from oocytes, normal oocytes, which have been injected with sperm RNA. sperm RNA coming either from control males or from traumatized males. So the idea is that if the sperm RNA contains a signal, contains a memory or alterations which are linked to trauma exposure, these animals and perhaps also their offspring should show signs of symptoms of a stress exposure. And this is indeed the case. You see here that the 4 swim tasks, so they show depressive-like behaviors and they show risk-taking behaviors. These animals here, but also their progeny, increased depressive-like phenotypes. Not just behavior was altered in these animals, but also the altered glucose level that I showed you earlier. Altered insulin level, but also glucose level. You see that when we stress them in this acute stress, there is not even a proper mounting of the glucose response. Glucose remains low all the time. So quite severe symptoms in these animals. They are indeed more severe than in the exposed males directly, because they happen very early on. You see the altered RNA populations, they are injected in the oocyte itself. So now the next question is that what could be the mechanism linking stress exposure to these alterations in microRNA or in the epigenome in germ cells? One common factor in between all organs, we have seen, as I said, we have seen alterations in brain, in blood, in heart, in germ cells, so in many different tissues. So a common thing across these tissues is blood. So imagine that if when the animal, a pup, is or a young boy or a young girl is exposed to trauma, blood composition is going to be perturbed. I mean, this is not new. It's known that you look at CRP, inflammation, for instance, or various proteins, metabolism is altered in people who are exposed to stress, and this has been confirmed in rodents. But the idea is that this altered blood composition, there are factors in these which are, or alterations which send, I mean, there are signals which reach testes and which give a signal to germ cells, and that these signals are then responsible for the perpetuation of the symptoms. So now the question is, what are these signals? So to approach this question, first what you need to do is to know what this blood composition is, how it is altered by stress. So what we did was to use mass spectrometry. We did unbiased high throughput metabolome, proteome, and transcriptome analysis. And what we observed, that was several years of work. I'm just showing some of the data in one table, but we did this so during the trauma itself and many months after the end of trauma and in the offspring. So here you see the result of a metabolomic analysis in plasma of pups, so 8 pups, mouse pups, which have been exposed to this trauma for 8 days. Then different pups that grew adults and the offspring of these adults. And what we observed quite consistently was an alteration of lipid metabolism. So while it was decreased, in particular fatty acid, polyunsaturated fatty acid metabolism, metabolism, so there are multiple factors, multiple components of this complex metabolic signaling pathways. So they seem to be decreased during the trauma, during the traumatic experience itself, but then there is a kind of a rebound and they are increased in adulthood. Bile acid biosynthesis is also altered and steroidogenesis. And some of these changes are also present in the offspring. So generally speaking, lipid metabolism in blood or in plasma of the mice exposed to trauma and the offspring is altered. So what does it mean? What could be the link? Is there any link between lipid in your blood and germ cells in testes? Yes, it could be actually, or at least the first question is there any link between fatty acids or lipids and chromatin and epigenetic factors, the epigenome? and then whether there is a link with the epigenome in germ cells. Yes, there is a link because fatty acids, they are ligands to a number of receptors, in particular PPAR receptors. So they are quite complex receptors, which are transcription factors. They act as transcription factors. So they bind the ligands, and they are transferred to the nucleus, and then they bind to the cognate sequence on the DNA, and they act as transcription factors, and they activate the transcription of target genes, which are different, various. There are many different PPAR target genes depending on the cell type and the tissue, but they play a very important role in metabolism, and this is known. So we thought perhaps these alterations here during postnatal, during development and in adulthood are responsible for changes in PPAR activity. First, we wanted to check whether in human we see comparable changes, similar to what I showed you before with mouse, with RNA in sperm. So Ali, I mentioned earlier, is here, and he also collected or assembled a cohort of children from a SOS Children's Village, also in Pakistan, and he selected these children. It's a cohort of it's a small cohort, it's about 100 children, including control children who are going to the same school, local school. And these children, they have lost their father, and they were placed by their mother into this orphanage. So it's fairly similar to the trauma that our mouse model is experiencing. And so blood was collected from these children as well as saliva, and we conducted exactly the same metabolomic alteration and metabolomic analysis as in mice. And you see that to some extent, fatty acids seem to be altered in serum and in saliva. So overall, not comparable, but overall, it indicates that lipid metabolism is altered also in the blood and in the saliva of children exposed to severe stress. Now, is it possible that these alterations in blood can lead to changes in germ cells? So to test this idea, to provide causal evidence, what we did was to extract blood, collect blood from traumatized mice. So we traumatize them when they are pups, then we wait until they are adult, we collect their blood, prepare serum. We do the same in control. So we have serum samples from males which were traumatized when they were young, serum samples from control males. And we inject this serum into control animals, control males. We inject it twice a week for 4 weeks. So it's a chronic intravenous injection of this serum, which here we know already is quite severely impaired. Sorry, I can turn on my light because yeah, it's getting dark. Sorry. Excuse me, probably better like this. So we inject the serum chronically, and then we breed these animals to generate an offspring. So the idea is that if the components that we know in serum are altered, if they reach testes, send a signal to testes, we should see some symptoms in these offspring. And indeed, that's what we saw. We did not see any behavior symptoms. We saw things, but they were not consistent with the behavior alterations observed in the mice, which were directly exposed to the trauma. But we could reproduce a decreased weight that we know occurs. Well, that's the data in MSUS. So sorry, in blue, that's the data in mice directly exposed, and in yellow, it's the data in the offspring of males which have been injected with the serum. So you see that there is a decrease in weight, and this glucose response is reproduced. This altered level of glucose is reproduced. So the offspring of males which were chronically injected with serum from males which were themselves traumatized, their sperm cells are altered. They keep a memory of this trauma. They receive signal from this injected serum in a way to transfer these symptoms to the progeny. So does this do these signals involve lipids and the PPAR receptor? We think yes, to some extent, because we can reproduce this transfer by activating PPAR. So what we did was, now we don't inject the serum anymore, we inject the PPAR agonist. So the idea is that if the injection with the serum, if the effect of the serum injection involve PPAR, if we mimic PPAR activation, we should see some similar effects. So we did this, so we injected now not serum, but we injected a PPAR agonist, tesaglitazar, twice a week for 4 weeks. And then we generated an offspring. And indeed, you see that in the tesa injected offspring, so the offspring of males which were chronically treated with these PPAR agonists, they have a decreased weight and a lower amount of glucose in their blood. Similarly to the MSUS mice, and similarly to the offspring of males which had been injected with serum, and not just them, but also their offspring. So it suggests that serum contains, that lipid metabolism, or altered, the lipid metabolites which are altered in serum, that they can communicate with germ cells, and that this communication route involves PPAR. Indeed, the question is, the next and last question is whether PPAR is relevant for germ cells. Yes, indeed, at least in these conditions, so it's in vitro conditions where we have spermatogonia cells. They are immortalized cells. They are a bit artificial, but it's very difficult to culture primary spermatogonia cells. So spermatogonia cells, they are the germ stem cells. So that's why we use these immortalized GC1 cells. So there are spermatogonia cells which are immortalized, and we transfected them with a PPAR reporter. So whenever PPAR is active, it will bind to this reporter and there will be luminescence. So then the cells, these transfected PPAR reporter transfected cells, we treated them with serum. So if serum contains, we know that the serum has an excess of lipids. So we expect the PPAR reporter to be activated. Now the question is whether this is possible in GC1 cells, which are spermatogenic cells. Indeed, we see that if we treat these cells with serum, we see that there is an increase in PPAR activity. So PPAR reporter is activated. So which means that in GC1 cells, the system works. And it's the same if we treat the cells with serum from tesa injected males. So males injected with a PPAR agonist, this drug. And indeed, if we look at the sperm of males which have been traumatized themselves, we see that there is more PPAR at the surface of the sperm. So indicative, which correlates with the, which fits with our observation that PPAR signaling is increased in the mice exposed to traumatic stress. So in conclusion, I showed you that small and long RNA are altered by postnatal trauma in germ cells, but it's also altered in brain, blood, and saliva. And in some cases, across generation. I did not show you the offspring data, but we know that some microRNA are also altered in brain and blood, not in sperm, of the offspring. RNA or the RNA population in sperm are causally linked to behavior and metabolic symptoms in the progeny, because we can recapitulate some symptoms by RNA injection of oocytes, and that circulating factors can also reproduce some of the trauma effects. And we believe that they implicate fatty acid signaling. So fatty acids metabolism, which is altered in blood, and these alterations can send signals to testis during development, because testis is quite permeable during early life, because there is not yet any testis blood testis barrier, which is quite tight in adulthood, but not yet during development. And I showed you that at least with the cohort that we have assembled, that there are comparable RNA and metabolic alterations in human subjects exposed to childhood trauma. So I'll finish with a picture of the lab. It's an old picture now. We have not been able to take a picture all together without masks. This is planned for November, but it's a nice picture because it shows people who have left since then, who have contributed to the results I showed. So first of all, Isabelle Weiss is the lady who 20 years ago developed the model and showed the transgenerational transmission. Tamara Franklin was in the lab about 10 years ago. She showed that DNA methylation was altered across generations. Cathy Gap here a little more recently. She showed the RNA alterations and did this RNA injection. And Ali did all the human analysis, and Gretchen van Steenwyk, who graduated 3 years ago, I think. She did all the serum and blood analysis and the injection. And that's the team. A few people are still with me. And Francesca Manuela is the person who is now generating all these cohorts. She has a very efficient strategy and protocol to accommodate everyone's need in terms of MSUS mice along the year. We have fantastic collaborators here and in the UK, and also in Pakistan. And I'd like to thank the schools and foundations who are supporting our research. And I thank you for your attention.